Mucosal immunity. Mucosal immunity is the immune response that protects the mucous membranes lining the gastrointestinal, respiratory, urogenital tracts, and other mucosa-associated surfaces, such as eyes, salivary glands, and mammary glands. These surfaces are the primary entry points for pathogens, so specialized immune defenses evolved here. Why mucosal immunity matters? Most pathogens, viruses, bacteria, parasites, enter the body through mucosal surfaces, respiratory tract, gastrointestinal tract, urogenital tract. Therefore, a strong mucosal immune response is the first barrier against infection. Vaccines that stimulate mucosal immunity can block infection at the entry point, before pathogens spread systemically. It is a combination of innate, physical, chemical, cellular barriers, and adaptive, antibody and T-cell mediated, immune mechanisms. Components of mucosal immunity. 1. Physical and chemical barriers, innate defense. Epithelial cells with tight junctions, prevent microbial invasion. Mucus layer, produced by goblet cells, traps microbes. Cilia in respiratory tract, sweep pathogens out. Enzymes like lysozyme in saliva and tears, degrade bacterial cell walls. Defensins, antimicrobial peptides, secreted by epithelial cells, kill bacteria, fungi, some viruses. 2. Mucosa-associated lymphoid tissue, MALT. Mucosal immune tissues are highly organized. Key sites. GALT, gut-associated lymphoid tissue, Peyer's patches, appendix. BALT, bronchus-associated lymphoid tissue. Bronchial tract. NALT, nasal-associated lymphoid tissue. Example: tonsils. These tissues contain immune cells that detect pathogens and initiate responses. 3. Specialized cells. M cells, microfold cells located in Peyer's patches, transport antigens from lumen to immune cells. Dendritic cells, DCS, capture antigens and present them to T and B cells. Intraepithelial lymphocytes, IELs, mostly T cells located within epithelium, kill infected cells directly. Macrophages and neutrophils, provide phagocytosis and inflammation. 4. Secretory immunoglobulin A, sIgA. The most important antibody in mucosal immunity. Produced by plasma cells in lamina propria. Transported across epithelial cells into mucus via poly-Ig receptor. Functions. Neutralizes toxins and pathogens. Prevents microbial adhesion to epithelial cells, immune exclusion. Provides protection without causing inflammation, important for tolerance. 5. Adaptive immune cells. CD4 plus T helper cells, especially Th17 and Treg, regulate immune balance. Cytotoxic CD8 plus T cells, kill infected mucosal epithelial cells. B cells/plasma cells, produce sIgA, IgM, IgG. 6. Cytokines and soluble mediators. IL-10, TGF-beta, promote tolerance, suppress unnecessary inflammation. IL-17, IL-22, strengthen epithelial defense and recruit neutrophils. Functions of mucosal immunity. Barrier protection, prevents pathogen entry. Immune exclusion, sIgA traps microbes in mucus, prevents adhesion. Immune elimination, activated T cells, macrophages, and K cells kill invaders. Tolerance to harmless antigens, food, commensal bacteria are tolerated to avoid chronic inflammation. Communication with systemic immunity, responses at mucosa can influence whole body immunity. How mucosal immunization works. Process. Vaccine delivery. Vaccine enters via oral, nasal, or other mucosal routes. Examples: oral drops, polio, rotavirus, nasal spray, influenza. Antigen uptake. Antigens cross mucosal barrier via M cells or are captured by dendritic cells. Antigen presentation. APCs activate T helper cells in mucosa-associated lymphoid tissue. B cell activation and class switching. T helper cells stimulate B cells to undergo IgA class switching. Plasma cells produce dimeric IgA. Secretory IgA formation. Dimeric IgA is transported through epithelial cells via polymeric Ig receptor, PIGR. Released into mucus as secretory IgA, sIgA. Protective response. sIgA prevents pathogen adherence and invasion. Cytotoxic T cells kill infected mucosal epithelial cells. Memory cells remain in mucosa for long-term defense. Vaccines targeting mucosal immunity. Oral vaccines stimulate gut mucosal immunity. Oral polio vaccine, OPV. Rotavirus vaccine. Oral cholera vaccine. Oral typhoid vaccine, Ty21A. Nasal vaccines stimulate respiratory mucosal immunity. Live attenuated influenza vaccine, nasal spray. Experimental nasal COVID-19 vaccines. Edible/plant-based vaccines, in research stage. Genetically modified foods like potatoes, bananas producing vaccine antigens. Advantages of mucosal vaccination. Needle-free, non-invasive, patient-friendly. Induces both mucosal, sIgA, and systemic, IgG, immunity. Can block infection at entry sites. Suitable for mass immunization campaigns. Challenges of mucosal vaccination. Antigens may be destroyed by stomach acid, enzymes, mucus clearance. Requires higher doses or adjuvants to enhance immune response. Variability in response due to gut microbiota, nutrition, or pre-existing infections. Limited number of licensed mucosal vaccines compared to injectable ones. Mucosal immunity is a specialized defense system at mucosal surfaces, combining physical barriers, innate immunity, adaptive responses, and unique secretory IgA to protect against pathogens while maintaining tolerance to non-harmful antigens.